Tariq?
Okay, we start with today's lecture, and today's lecture is about immunomodulators. And in the immunomodulators, we will basically discuss mainly the immunosuppressant drugs and their relation with the transplantation. So your main topic is immunomodulator, but in the immunomodulator, the subtopic which we will focus today is immunosuppressants. Okay. First of all, we will start with the definition. What is an immunomodulator? So we define immunomodulator that these are the chemicals or the substance. They can either stimulate the immune system or they can suppress this system. This is why you are using the word modulator. So both things can be happen. Either they can stimulate or they can suppress any of the immune system complex. So we can say that immunomodulators are basically divided into two types. One is immunosuppressants that we will discuss mainly today, and it is also include immunostimulants as well. But today's talk is more focused on immunosuppression because they are clinically related with the transplant process. But in a broad spectrum, you should remember that immunomodulator also includes the immunostimulant as well. Okay. In the beginning, I would like to tell you that what are the different sites or what are the different regions where an immunomodulator can affect. The different sites or their mechanism, they could be related in antigen recognition and phagocytosis, one of them. Lymphocyte proliferation, this is the main part, lymphocyte proliferation or its differentiation. Synthesis of antibodies, which is mainly a function of B lymphocytes. Antigen-antibody interaction, the release of mediators due to immune response, and the modification of the target tissue response. Mainly you can remember or you can focus that the main player of the immune system are lymphocytes. So the drugs mostly that we will discuss today, they are also related with the lymphocyte, with their suppression and the different cytokines that are released from the lymphocytes. So the main focus of today's talk and related with the immunosuppressant is basically the lymphocytes. What are the clinical applications? Why we are discussing this topic? That therapeutically or clinically, there are certain diseases that are related with this immune modulation system or immune suppressant system. For example, we have diseases like myasthenia gravis. This is a disease of acetylcholine receptors. So the autoantibodies are formed. So patient have a problem with this movement or the muscles problem. Type 1 diabetes is also example of the autoimmune disorder. Rheumatoid arthritis is also there. So there are many diseases in which there is immune system is basically exaggerated or it is overstimulated. So you need to suppress down the immune system. So many clinically important. And today's talk, as you see in the topic as well, that transplantation process is also very important with the immunosuppression. So the transplant, you need to suppress the immune system. So bone marrow transplant, kidney transplant, so any sort of transplant, in that you require suppression of the immune system. And we will discuss in this scenario today. So this is the clinical application of immunomodulator or immunosuppressant mainly. Okay, so we have focused on in the immunomodulator. We now come to the topic of immunosuppressant. So this is basically the main topic of the today. In the first slide, I would like to give you an overview of what are the different drugs that we will discuss today. So immunosuppressant can be classified according to their mechanism. For example, The first category, or the main drug of this immunosuppressant class, is T cell inhibitor. I told you that T lymphocytes are the main player of the immune system. So the specific T cell inhibitors are the key drugs for the immunosuppressant. In these drugs, the examples are cyclosporine, this is the main drug, tacrolimus, sirolimus. So we will go in details of each drug, their mechanism, their side effects, all this. The first class is specific T cell inhibitors that basically act on T cell activity. The second class we have the cytotoxic drugs. Cytotoxic drugs are basically the anti-cancer. They can also be used in the immunosuppressant because cytotoxic drugs are basically cell-killing drugs. So we use these drugs to suppress the immune system or T lymphocytes. So they are the killer of the T lymphocytes. So we also include these drugs in immunosuppression, although they have a separate class. They are basically discussed in anti-cancer drugs, but here we include in the immunosuppressant drug. Third drugs are glucocorticoids. You know very well the glucocorticoids are the anti-inflammatory drugs, and inflammation is also a sort of immune response. So glucocorticoids, they are also somehow related, or they are basically, if we say more precisely, they are the helping cells, or they are the helping drugs for the immunosuppressant. They help these drugs, the main drugs like T cell inhibitors and the cytotoxics, they help out. So they are used in combination. They are not the sole agents or only agents, but they are also used in the combination. And obviously we have the antibodies, which is a new treatment. Specifically, you target the drug. You target the cells like T cells. So you have antibodies like monoclonal antibody, polyclonal antibody. Immunomodulator is an example of monoclonal antibody. Antithyroidglobulin is a polyclonal antibody. So antibodies are also there. So the first slide, this will come again to just memorize that they are the basically main classification of the immunosuppressant. And now we will go in detail one by one of each drugs. For example, we start with the cyclosporine, the main drug. Cyclosporine is basically chemically it's a peptide, and it is extracted from a soil fungus. What it is basically inhibits the proliferation of the T lymphocytes. So again, T lymphocytes, and in the T lymphocytes, the T lymphocyte release many cytokines, so the chemical mediators for the system. So in the T lymphocyte, interleukin-2 is the main player related with the immune system. So they basically inhibit the release of interleukin-2 mainly. So you will come across with this again and again. The interleukin-2 is the main cytokine that is involved in the immune system, and we will see the different drugs that will inhibit this the production of this interleukin. They arrest these lymphocytes in G0 to G1 phase. You know, this is the phases of the cell cycle. So all these drugs do that, it inhibits the proliferation of the T lymphocytes and mainly inhibit the release of the interleukin-2. Specifically, if we say, if we go in detail of the mechanism of the drug, we say that these drugs are basically can be classified as calcineurin inhibitor. What is a calcineurin? It's a protein present into the T lymphocytes, and this calcineurin is responsible for. So calcineurin, the drug cyclosporine first goes into the T lymphocytes, and there's a protein known as cyclophilin. So it first binds to the cyclophilin, and then they make a complex, cyclosporine plus cyclophilin. This is a complex. This is formed when cyclosporine combines with the cyclophilin. Complex is formed, and this complex basically inhibits the calcineurin. You can better be understand diagrammatically you can easily have this concept. See this cyclophilin, this is the drug, and now this is first bind to the cyclosporine is the drug, and this is basically binding to cyclophilin, the first protein, and these two drug make a complex, and then they finally inhibit this calcineurin. Then this calcineurin, once inhibited, the cyclic process goes on, and finally there is no production of the interleukin 2, which is our target molecule. So you can remember that cyclosporine is a calcineurin inhibitor, because we have also other drugs that inhibit the calcineurin, which is a key molecule to produce the interleukin 2. Okay, clinical use, as I described earlier, that we are discussing this drug, immunosuppressant, for the in the scenario of transplant. Transplant, whenever you have transplant, you need to suppress the immune system for the to avoid the rejection of the transplant. So transplant rejection, we use the cyclosporine. Usually this drug is given 12 hours before the transplant in order to have the suppressed immune system. Along with transplantation, they can be used for autoimmune disease. I just discussed, like you have rheumatoid arthritis, and you have also autoimmune disease like thyroiditis or the diabetes mellitus. So the main use of this drug as a to avoid the rejection of the transplant, but they can also use for the other autoimmune disorders. Every drug has side effects, so this drug also has a first side effect. The main side effect is the suppression of the immune system, which is also the patient is liable to any infection because you're suppressing the immune system. But always we have to see the risks and benefits. In transplant patient, we cannot take the risk to have an active immune system, so we suppress it. Other side effects than this immune suppression, we have the renal dysfunction, the hypertension can be there, hyperplasia is one of the side effects. These drugs can also be diabetogenic if they are used with the glucocorticoids. So always you have side effects of every drug. We always try to focus the main side effect that you should remember. So I just highlighted boldly: renal dysfunction, hypertension, hyperplasia, and the diabetogenic. Okay, so this is about cyclosporine. Another drug, the second drug we are discussing is the tacrolimus. Tacrolimus is also classified as calcineurin inhibitor. So the first drug is cyclosporine. This is also a calcineurin inhibitor, and the second drug is tacrolimus. This is also calcineurin. So if you know that calcineurin inhibitor, no need to repeat the mechanism. I just discussed this. It has the same mechanism that that it first binds to the cyclophilin, and then it releases the calcineurin. The other part is that chemically it's a macrolide. Macrolide, it's a class of antibiotic that you have discussed. Like you have a drug like azithromycin, clarithromycin. They are the class of macrolide. So this also belongs chemically belongs to the class of macrolide. But here this mechanism is towards the calcineurin. So it's an additional part. Although macrolide has a different, they are protein synthesis inhibitor, but this drug is effectively or has an additional function that inhibits the calcineurin. Obviously, when it inhibits the calcineurin, the production of IL-2 or interleukin 2 is reduced. Important thing is that the tacrolimus is 100 times more potent than the cyclosporine. So the first drug that came into the market was cyclosporine, but then we have the tacrolimus, which is more potent, 100 times more potent than the cyclosporine. This drug is orally as well as infusible. This is pharmacokinetic, and efficacy is high. Okay, so second drug is tacrolimus. The third drug is sirolimus. Sirolimus, the name are similar, but the mechanism is different. This you can say in one word that this is an mTOR inhibitor. Okay, what is mTOR? mTOR is basically a mammalian target of rapamycin. It's a protein. So rather than it affects on calcineurin, it is also affecting on T lymphocytes. But the target of the intercellular target of this drug is not calcineurin, but another protein that is known as mammalian target of rapamycin. Okay, so all you need to remember that they are also T cell inhibitor, but rather than the target of calcineurin, their target is mTOR. So the first two drugs we discussed, cyclosporine and we have these tacrolimus. They are calcineurin inhibitors. The sirolimus is a T cell inhibitor, but it is an mTOR, mammalian target receptor inhibitor. mTOR is a serine kinase involved in many cell cycles. This is the detail of mTOR. It's an enzyme or protein, you may say. This drug is only available as oral drug. So, and this is good that this you can give this drug orally. Oral drugs are easy to take, and you can give this drug so that the patient can take at home. And again, the same process that they also inhibit the interleukin 2 production. And more precisely, that they inhibit the cellular response to interleukin 2. So it's a little modification that they not only suppress the production, but they also decrease the response of desensitization of the T lymphocyte for the interleukin 2 response. Okay, so third drug is sirolimus. So I can repeat again. We discussed the first class, that is the specific T cell inhibitor, and in this class we discussed the cyclosporine, calcineurin inhibitor, tacrolimus, again a calcineurin inhibitor, sirolimus. This is an mTOR inhibitor, but all effects on T cells, and they inhibit the proliferation or they inhibit the release of interleukin 2. Second drug that we are going to discuss is cytotoxic drugs. This is a repeat slide just to memorize. Now we come to the second class, that is the cytotoxic drugs, and we will discuss these drugs one by one. Cytotoxic drugs, I already told you that basically they are anti-cancerous drugs. And what do you mean a cytotoxic, a drug which basically either they basically kill the cells. So in the cancer, you know, there's a rapid proliferation of the cells. So all you need to do is to kill these growing cells. So these are very effective drugs, and they are very potent drugs. So we use this drug in a sense that we are controlling the activity of T lymphocytes here, because the target is to suppress the immune system and to suppress the T lymphocytes. First drug is azathioprine. It's a purine antimetabolite. What is purine? Purine, you know, that's a part of part of the DNA molecule. You have the purine basis, you have the pyrimidine basis. So they bind to the purine basis, and they inhibit the transcription process or the other process. So they are the purine antimetabolite. You all have to remember a one-line sentence for their mechanism of action. So the purine antimetabolite, and they specifically selectively affects the cytolytic lymphocytes or T lymphocytes. Again, they have a more attention toward the lymphocytes. Although always remember that the cytotoxic drugs are not very specific drugs. They are not target-oriented drugs. Generally, they kill all the cells because they have a numerous effect. But azathioprine here we can use in order to avoid the proliferation of the T lymphocyte, because T lymphocyte, when it will be inhibited, there will be no cytokine release. Graft reject, always the use is for graft rejection or transplant. This drug is less effective than cyclosporine, because cyclosporine is more effective primary drug, but still we use this azathioprine as a second line drug or second choice of the drug. And this is also used in combination usually with the corticosteroid, we combine with the corticosteroid. The second drug with cytotoxics or anti-proliferative drug, the proliferation I told you, is a multiplying of the cells. So cytotoxic drugs are also known as anti-proliferative drugs. Cyclophosphamide, well-known anti-cancer drugs, is an alkylating agent. So cytotoxic drug with anti-cancer drugs can be classified differently. Antimetabolite, alkylating, this is sort of classification. I just cited one word so that whenever you will be recalled, you should know that what is the specific mechanism of this anti-proliferative drug. So alkylating agents are the drugs that basically make a chemical bonding with this DNA, and they block this DNA. So these sort of drugs are known as alkylating. Alkylation is a chemical reaction. So these drugs basically alkylate, or they basically bind with the chemically with the DNA, and they block. They cause intercalation, or whatever the word you can use. But the target is DNA, and they block their activity, so there will be no transcription. Alkylating agent more effect on B cells. B cells are known for their antibodies, so they are more precisely used with the bone marrow transplantation, where B cells have an activity. So again, this is a class you can use as an immunosuppressant. The third drug as an anti-proliferator, as a cytotoxic drug, is methotrexate. This is very well known. You have discussed this drug in different topics, like in autoimmune disorder, we use this drug, methotrexate. The mechanism is folate antagonist, that it's basically effect on folate. Folate is required for the maturation of the DNA. So once you inhibit the folate activity, the DNA will not be matured, and there will be certainly the killing of the cells. So this drug is not very effective for the transplant process, but this is a very good drug for autoimmune disease. Autoimmune is we can use, but this is also a choice for transplant as well. So folate antagonists inhibit the production, even if it inhibits the maturation of the cells. So T lymphocytes will not be mature, and finally will not have the cytokine release. Okay, fourth drug regarding with the cytotoxic drug or anti-proliferative drug is mycophenolate mofetil. This is a drug. How it works? It inhibits an enzyme known as inosine monophosphate dehydrogenase, and this enzyme is essential for the synthesis of GTP in the T and B lymphocytes. So the target is an enzyme, inosine monophosphate dehydrogenase, and it basically inhibits the synthesis of the T and B lymphocytes. Again, once you inhibit this lymphocyte, antibody function will be also reduced because of B cells, and this drug has a successful role in renal transplantation, and this is a good drug you can use. It also reduces the dose of cyclosporine, always a standard of regimen. So we always compare the drug with the cyclosporine, which is a primary drug, the prototype of drug for the immunosuppressant. Always we give you, we repeat this word to our in lectures, that remember always the prototype of drug. Whenever you go, you are asked for the Viva or anywhere, the thing that you should not miss is the prototype of drug. For example, if we talk about immunosuppressant, the prototype drug is cyclosporine. We talk about the NSAID, the immunotype of drug, the prototype of drug is aspirin. So never miss the prototype of drug. So we compare also this drug. So it reduces the dose of cyclosporine toxicity, and it's an alternative choice, or they can be given along with this cyclosporine. So the side effects are leukopenia and other things. So this is again a repetition what we have discussed up till now. We have discussed specific T cell immunity. They are the main players, in which the cyclosporine is the main drug. The tacrolimus or sirolimus are the new drugs, more potent, less side effects, modified form for the cyclosporine. Then we use cytotoxic drugs, so they are also known as anti-proliferative drugs, in which we just discussed the azathioprine, superior antimetabolite, cyclophosphamide is an alkylating agent, methotrexate is a folate antagonist, but we didn't discuss this chlorambucil. For the sake of the time, we cannot discuss each and every drug. Mycophenolate mofetil, sorry, and this is the monosine we just discussed the mechanism. Now we come with the glucocorticoids, then we'll discuss the antibodies. Glucocorticoids, you already know that they are the potent anti-inflammatory drugs, and they also have an immune suppression as well, because they inhibit all the inflammatory mediators they effects on prostaglandin, they effects on leukotrienes. So they are immunosuppressants as well as they are anti-inflammatory. They also affects on interleukin production, cytokine release. So they also regulate these cytokine genes. Or the steroids, their main mechanism is that they goes into the genes, and then they have a slow response. So they inhibit the cytokine genes, and finally they stop progression of the release. So they are short-lived, rapid lymphopenic effects. So they reduce the lymphocyte counts, and they are good companion drugs. So they are not a main player, but they could be used as a companion drug with the main drug like cyclosporine. Again, I told you that we will use this word again and again. So you can have a combination of cyclosporine with this steroid. This will help out in the immunosuppression. The mechanism is no need to discuss because we've already been discussed this mechanism in the other lecture as well. Cytotoxicity, everybody knows that they cause bruise reduction, the weakening of the bone, risk of infection, or the cataract, hyperglycemia. Long terms are the side effects of the steroids. Okay, so the last part of this talk about is the antibodies. What are the different antibodies? Because the current management of many we are focusing on target drugs, or target oriented pharmacology is very common, and in which we have the antibodies. For example, you have the first antibody we are discussing is the muromonab. What is a muromonab? Murine is a word used for the mice or the rat, mainly for the rodents. So murine monoclonal antibody, this is derived from the mice, and chemically or specifically it's a CD3 protein complex inhibitor. What is CD3? CD3 is a sort of protein that is present on the surface of the T cells. So it's a T cell co-receptor. CD3 is a T cell co-receptor, so it basically inhibits this T cell co-receptor. So ultimately, the result is that the T cells are not activated and they are not stimulated to produce any plant production. This is a drug used for acute organ transplant rejection, like in renal, cardiac, and hepatic allografts, and it also prevents the nephro and hepatic toxicity of cycles. So good alternative drug for cyclosporine, and the specific mechanism is on CD3. Right? Also, you can remember with this diagram that CD3 is a monoclonal antibody that basically causes the rapid internalization of the T cell receptor. I told you that CD3 is a co-receptor that's present on the T cells. So they got the internalization of this receptor, so antigen will not bind it. So when there would be no antigen antibody reaction, there will be no stimulation of the T lymphocyte. So they got the internalization of the T cell co-receptor, CD3 receptor, and finally antigen is unable to recognize the T cells, and there will be no reaction. Toxicity, antibodies are good in action, but again, they can also produce some toxicity. Toxicity of this include the flu-like symptom. We also call it cytokine release syndrome, which has a high fever, chills, headache, tremor. So this is a short condition. Sometimes, if you give, the doses are very important when you are giving with the antibodies. You have to be very specified with the dose, because the small increase in the dose because serious toxicity. So high dose may be related to the life-threatening condition. Otherwise, if they are given in a certain range, they do not cause much effect. So cytokine release syndrome we also know. The second antibody we use is basically interleukin-2 antagonist. So they basically directly affect on interleukin-2. And the drugs we have the basiliximab and daclizumab. They are the two human monoclonal antibody, means they are derived from the humans, and they are used as a prophylactic treatment of in the acute transplant or renal transplant rejection. So they are the interleukin-2 inhibitor, basiliximab and daclizumab. Specifically, they bind to the CD25 antibody, and ultimately they affect the interleukin. All you remember is that they are the interleukin-2 blocker and interleukin-2 receptor inhibitor. Aciliximab is 10 times more potent than daclizumab. The potency of aciliximab is more, but the again, and the half-life is also high, 7 days. Daclizumab has a 20 days half-life. So potency-wise, they have a difference. Otherwise, the target is the same. Another antibody we use is an antithyroidglobulin. This is a polyclonal antibody. It's not a monoclonal antibody. It's a polyclonal antibody, and this is derived from the horse or rabbit. And this antibody basically affects on CD8 cells. So you know the T lymphocytes are classified as CD4s, different, right? So they are effective on CD8 positive cells, and they are used, currently used for acute allograft rejection. So this is another antibody we use. This is not a monoclonal; it's a polyclonal antibody. This drug also have a side effect known as serum sickness, so it may cause anaphylaxis. So this is also a acute reaction. It is less expensive than Immunomodulator and others. So this is used because it's cost effective. Otherwise, the antibodies are very high. Cost is very high. Treatment is good, but the cost is very high, and we also take care of the reaction as well. This is about the, this is the last slide regarding the individual drugs. Now we can summarize in one go that if you want to remember the drugs with their specific action, we discussed that immunosuppressant, today's topic is mainly immunosuppressant. So the main drug is the specific T cell inhibitor. We discussed cyclosporine, tacrolimus, sirolimus. Cyclosporine, tacrolimus, they are the calcineurin inhibitors. Sirolimus is mTOR inhibitor. They are the specific T cell inhibitor, the main player, first line agent. Then we can use this cytotoxic drugs, or we can say that anti-proliferative drugs, the anti-cancer drugs. We discuss the azathioprine. It's a nucleotide inhibitor or false nucleotide incorporation. Then we have mycophenolate mofetil, inosine monophosphate dehydrogenase inhibitor. Also, we discussed the cyclophosphamide, methotrexate. Then we have the antibodies. The antibodies, we have Immunomodulator, monoclonal antibody that is effective against CD3 cells. Daclizumab and aciliximab, they are effective on interleukin-2 receptor. They are also monoclonal antibody. Then we discussed antithyroidglobulin. This is a polyclonal antibody, and that affects on CD8 cells. Lastly, we discussed the glucocorticoid, which are not the main player, not the main drugs, but they can use as a combination therapy because they have an effect that they depress the immune system somehow. So they can also regulate the gene transcription. So they are the co-player with this immunosuppression. This is a summary. This slide can help you out when I have a point of view that you can remember in one go what are the different drugs as an immunosuppressant. That is the last slide. Thank you very much. Now the session is open for your questions. Chat. We have a first question that is calcineurin also present in neurons to the same